Hello, everyone, and welcome to today's webinar, Smarter Sample Preparation for Single-Cell Sequencing. I am Matt Ashton of Labroots, and I'll be your moderator for today's event. Today's educational web seminar is presented by Labroots and brought to you by Miltenyi Biotech. To learn more, visit miltenyibiotech.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. I'd like to now welcome our speaker, Carina Emery, Product Manager for Sample Preparation at Miltenyi Biotech. Carina, you may now begin your presentation.
Hi, everyone. Thank you for tuning in today. We have a lot of material to cover, so let's just dive right into it. So first we'll start out today with a brief introduction to Miltenyi Biotech, and then we'll take a really deep dive into tissue dissociation methods. So we'll look at standard methods and then modifications of tissue dissociation methods that have really been tailored towards single-cell sequencing and really preserving that in vivo gene expression pattern. Then we'll look at nuclei isolation methods, which has also really increased in popularity lately. And we'll also look at some of the differences between cellular composition you might get between cells and nuclei. And then finally, I'll summarize everything and give some useful tips. Miltenyi Biotech was founded over 30 years ago by Stefan Miltenyi based on his invention of MACS technology, which enables the fast and gentle magnetic separation of specific cell populations based on their surface antigens. Early on, Miltenyi recognized the importance of developing research products that avoid cell stress and maintain the cell's native functional states. Or, in other words, we understand that good research starts with happy cells. Today, Miltenyi Biotech has grown into a global company with a wide variety of products to support anything from basic research to cell and gene therapy. Our comprehensive portfolio includes solutions for sample preparation, cell isolation, cell culture, flow cytometry, and advanced microscopy, so a pretty broad portfolio. However, today's talk will really focus on our solution for sample preparation. When you work with solid tissue, there are a number of steps to complete before you have a single cell or nuclei suspension ready for single cell sequencing. You might need to store your tissue prior to processing. Then you need to dissociate your tissue into a single cell or nucleus suspension. And then followed by that, you need to clean up your suspension to remove debris and dead cells. As an optional step, you can isolate target cells via magnetic cell separation or multi-parameter cell sorting. Finally, the last step prior to proceeding with your downstream assay is the QC of your cell or nuclei suspensions to ensure sample quality and the correct concentration. I wish we had time to go over all of these steps today because they're all important, but I will mainly be focusing on tissue dissociation. So to learn more about the other steps, we have a really great webpage focused on our solutions for preparing cells and nuclei for genomic applications that I highly encourage you to check out. Tissue dissociation is the process of transforming solid tissue into a single cell suspension. For our purposes today, I'm also going to call it dissociation if you're transforming solid tissue into a single nucleus suspension. All of these methods traditionally involve manual mechanical processing, and for cell dissociation, it may or may not include enzymatic digestion. So for example, you might use a stick blender to process your sample, so that might be semi-automated. Or some people use bead beaters to dissociate their tissue samples. I do not recommend that. That is far too harsh on your cells. If you have particularly soft tissue like spleen, you might be able to get away with just mushing it through a cell strainer. And probably the most common method that people use when they're doing manual dissociation is they'll just take scissors or razor blade and just chop up the tissue into little pieces. Simple as that. So all these methods work, but Molteni uses something a little different. Since 2008, Miltenyi Biotech has offered gentleMACS technology as an automated solution for tissue dissociation. gentleMACS technology takes advantage of simultaneous mechanical disruption and enzymatic digestion to give the most gentle dissociation possible. Mild mechanical shearing disrupts the tissue and increases the surface of the tissue so that enzymatic incubation time can be reduced to a minimum. In addition, enzymes reduce the need for strong mechanical forces because they soften the tissue by digesting the extracellular matrix and the adhesion molecules. So, dissociating tissue and preparing your cell suspension without introducing bias is a really critical step for any single cell sequencing experiment, but it's a really tough undertaking, especially when isolating cells from complex tissues such as brain, kidney, or lung. Ideally, when you're making a single cell suspension, your goals are to have a high viability cell suspension free of debris or cell aggregates. Likewise, if you're working with nuclei, you want it free of debris and cell aggregates, but instead of high viability, you're looking for nice intact nuclei. And then here's the hard part. You also want to make sure you're maintaining the in vivo expression profiles of your cells as well as maintaining the in vivo cellular composition. So while tissue dissociation methods have been around for a long time, it's really only been in the last several years that there has been an invested interest and ability to understand and document the impacts of tissue dissociation on gene expression, which of course is the result of the rising popularity of single cell sequencing. The graph on the left shows the tremendous growth in the number of publications with single-cell sequencing experiments, while the graph on the right shows the total number of cells analyzed. This explosion of single-cell sequencing data on a wide variety of tissues and organs over a very short period of time have left some to question how faithfully this data represents the cellular compositions and gene expression profiles of tissue in vivo. The first publications to explore this question came in 2017 when this technology was just starting to take off. In an early single-cell RNA-seq experiment of dissociated and FACS-sorted muscle stem cells, the authors discovered that a subset of cells express high levels of stress-associated immediate early genes, including FOS. They performed single-molecule RNA FISH to demonstrate that immediate early gene expression is an artifact of dissociation. In another paper published the same year, it was shown that stress-associated immediate early genes such as FOSB and Jun were significantly upregulated in kidney when a traditional warm dissociation protocol was compared to a cold dissociation protocol, where the cells were maintained below 6 degrees to theoretically freeze the in vivo gene expression profile. These publications provided clear evidence for what many researchers had long suspected, which is that dissociating tissue and thereby removing cells from their native microenvironment will cause gene expression alterations in some cell populations. So now the question becomes, what are the implications of dissociation-induced gene expression alterations, and what do we do about it? This question was tackled in a really great review paper by Machado et al., where they summarized what we know about the impact of dissociation-induced gene expression artifacts in single-cell sequencing experiments, as well as emerging methods to mitigate these alterations. One of the things they did that I really liked is that they point out that not all types of experiments are impacted equally by dissociation-induced gene expression artifacts. For example, it is least impactful in experiments that compare cells at the same state but different conditions. The assumption is that the dissociation-induced artifact is equivalent between conditions and will be canceled out. Next, it could have an impact when examining cellular heterogeneity, such as building a cell atlas. We'll go into some of the differences in acquiring different cell type compositions from different methods later on, and rare cell populations may be lost based on your dissociation method. And while these atlases are typically built with the goal of documenting healthy tissue in a resting state, dissociation-induced artifacts could confound that. Finally, the experiments most impacted would be those comparing cells at different transition points in a dynamic process. For example, experiments interrogating activation of quiescent muscle stem cells in response to injury would be very impacted by this, and the authors even go into examples of how it did impact their research. In a short time span, a variety of methods have been proposed to tackle the issue of dissociation-induced artifacts, and Machado et al. Make this really nice summary table. Starting from the right side of the table, bioinformatic methods are frequently proposed to correct sample preparation artifacts, and some studies have removed cells expressing high stress signatures, while other studies have completely removed panels of stress-related genes from their analysis, and others have implicated even more complex algorithms. So these post hoc data correction methods come at the risk of masking real biological variability and generally should be used as a last resort. Next is spatial transcriptomics, which has immense potential and really deserves its own presentation, but we're not going to cover that today. It's still really in an early stage and is not widely and easily accessible to all. Next is RNA tagging, such as TRAP-seq, and this method enables studying cell type specific RNA without the need for dissociation or cell isolation. However, this method requires sophisticated transgenic animal models. So I'm going to spend most of my time covering these last four methods. And one method that I'm very excited about is fixation. So methanol has been used for a while and because it's compatible with most single cell sequencing chemistries. However, it's been used with variable success. And one thing to note is that most investigators are typically fixing cell suspensions in methanol, not tissue. So obviously that does not do anything to prevent dissociation artifacts. Really, the biggest weakness of tissue fixation with PFA was that until very recently, it was not compatible with most single cell sequencing chemistries or 10x genomics. However, that changed. Very recently, 10x Genomics launched their chromium-fixed RNA profiling kit, which is compatible with formaldehyde-fixed human tissue. What makes this product especially powerful is that it's compatible with fixed tissue rather than just fixed cell suspensions. This facilitates tissue preservation at the clinical site and is a really simple and effective way to lock in your cell states as they were in the intact tissue. Along with the kit, 10x released a demonstrated protocol for dissociating fixed tissue using the gentleMACS octo dissociator. They even reported promising results using this kit with FFPE tissue that was dissociated using Miltenyi's FFPE tissue dissociation kit. So this is all very new, and Miltenyi Biotech has not had the chance to develop a validated fixed tissue dissociation protocol, but we are working on it. So I'm really excited to see how the scientific community takes advantage of this technological advancement. But for now, we'll have to move on to more established methods. Cold tissue digestion is an established method in that it was initially published on 5 years ago, so a long time for single cell sequencing methods. The transcriptional machinery is largely inactive at low temperatures, which slows down the dissociation-induced gene expression alterations, and the enzyme most commonly used for cold digestion is a cold active protease derived from Bacillus licheniforms, which has a history of use in industrial applications and in laundry detergent. You can also use standard digestion enzymes at 4 degree, but of course they're going to be less effective. A big benefit of cold digestion is that you end up with viable cells that can be used for any other method, like cell sorting. Downside of cold digestion is that transcriptional changes, maybe like cold shock response, are still possible. And the biggest problem I see with cold digestion is just simply protocol development. You can't just take a warm dissociation protocol, perform it at 4 degrees, and expect the same cell yields. So why don't we take a look at an example of dissociating the same tissue at 37 degree and 4 degree, take a look at the two different protocols and the results. This was a very thorough methods paper that came out of a group from Australia, and it looked at the effects of a variety of different sample prep methods for single cell sequencing. So the figure shows all the methods they tested, but right now we're just going to look at their comparison of warm versus cold dissociation. First, let's take a look at the dissociation protocols. So for warm dissociation, they use the Miltenyi Biotech multi-tissue dissociation kit 2 with our gentleMACS octo dissociator. It's a 45-minute protocol. To prepare the tissue for that protocol, you simply cut the fresh kidney into quarters, and then you filter, wash, and filter, or then what they did was they filtered, washed, and filtered again. So compare that to the cold dissociation protocol, where first they had to mix the fresh mouse kidneys on ice into a smooth paste, and then they transferred that kidney paste into the gentleMACS C tube, which also works on dissociator with their own enzymatic solution containing the cold active protease. Then they had to combine tertiation with the run on the gentleMACS in the cold room. And so they did this back and forth, and that lasted about 40 minutes. And again, they filtered, washed, and filtered again. So both of these protocols ended up taking about the same amount of time, but one required the cold dissociation, obviously required significantly more mechanical disruption in order to get a single cell suspension. The authors did not comment on total cell yield in the publication between these two different protocols, but in Miltenyi's experience, effective enzymes are a really important part of attaining good yield. And it's really unfortunate that a lot of people don't publish their cell yields when they publish methods. But typically with cold dissociation, I think it's fair to expect a lower cell yield than you would get with warm dissociation. However, a longer protocol and potentially lower cell yields may be a fair trade-off to prevent dissociation-induced artifacts. The authors examined a signature of 17 stress response genes and found that expression was higher in cells that went through warm dissociation compared to cold dissociation. They also observed that the stress response varied by cell type, which was quite interesting. Some of the cells showed a significant upregulation of stress genes during warm dissociation, while other cell types showed no stress response. Another promising method to prevent dissociation-induced artifacts is to add inhibitors to the digestion mixture that will stop transcription or translation. Actinomycin D has been demonstrated to be effective in several studies. Benefit of using inhibitors is that it maintains cell viability and is easy to integrate into existing dissociation methods. The main disadvantage is that this is still a relatively new method, and more studies are needed to explore off-target effects and the impact of not inhibiting all mechanisms of gene expression alterations, such as gene repression and post-transcriptional modifications. Microglia are a good cell model for dissociation-induced artifacts because they're highly reactive to their local microenvironment. Microglia are the resident immune cells of the brain, and microglia activation has been implicated in aging and disease, so examining microglial cell states is of great research interest. So here's another thorough method paper that looks at a number of dissociation conditions for adult mouse brain. They performed single-cell RNA-Seq on fact-sorted microglia that they obtained through either warm dissociation with Miltenyi Biotech's adult brain dissociation kit used on the gentleMACS octodissociator, or a cold dissociation protocol that relied on a Downks homogenizer without the use of enzymes. And both methods were tested with and without the addition of transcriptional and translational inhibitors. The authors were motivated to find an enzymatic protocol with minimal ex vivo transcriptional alterations in order to obtain higher yields, particularly when working with adult mouse brain. Enzymatic digestion without inhibitors resulted in a significant cluster of microglia that the authors designated as ex vivo activated microglia. They did additional analysis to demonstrate that the activation was mainly occurring during the dissociation step. So the activated microglia were characterized by increased expression of immediate early genes, and importantly, they showed that the cluster of ex vivo activated microglia was eliminated when inhibitors were included in the enzymatic digestion, which shows great promise for this method. Finally, let's take a close look at using single nucleus RNA-Seq as a substitute for single cell RNA-Seq. Single nucleus RNA-Seq has gained significant popularity because it is the only method compatible with flash frozen tissue and large cell types. Isolating nuclei is also very fast and can be performed on ice, which makes it ideal for single cell sequencing. The main downside of using nuclei is that you lose all the cytoplasmic RNA and the cell surface markers that are typically used for cell sorting. Investigators new to single-cell sequencing are often concerned that their analysis will suffer from analyzing nuclear rather than whole cell RNA, but several studies have shown that you can detect similar number of genes and transcripts and get comparable information from nuclei if intronic reads are included in your analysis. Depending on your starting material and your desired analysis, you may not always have a choice to use either cells or nuclei. So, for example, clearly feature barcoding of cell surface markers will not be compatible with nuclei, and nuclei are also not recommended for TCR or BCR sequencing or analyzing RNA splice variants. On the other hand, nuclei are required when performing ATAC-seq or when using the 10x multiome kit. Your starting material may also limit whether you're able to use cells or nuclei. For example, if you have flash frozen tissue, then your only option is to sequence the nuclei since it's not possible to obtain viable cells from flash frozen tissue. Or if you're interested in analyzing very large or fragile cells that do not survive the dissociation process or perhaps are too large for the microfluidics of your single cell sequencing platform, then nuclei are the best option. One drawback of using nuclei is that they are very difficult to isolate specific types of nuclei. So if you're working with a transgenic animal model, expressing a fluorescent marker in your cell type of interest is possible. If not, you have to rely on immunolabeling to enrich nuclei, and it's very difficult. Miltenyi Biotech offers a great solution for automated nuclei extraction on the gentleMACS octodissociator. It combines gentle detergent-based cell lysis with mechanical disaggregation provided by the C-tube, which is controlled by the instrument. The whole protocol is performed cold, and later this year, we'll be releasing cooling units to go with the gentleMACS Octo to further simplify this workflow. Here are the steps of our nuclei isolation workflow. You can start with up to 200 milligrams of tissue and then drop either fresh or frozen tissue into a C-tube with chilled nuclei extraction buffer. The C-tube is then closed and placed onto the gentleMACS instrument where tissue lysis and homogenation is completed in only 5 minutes. Moreover, up to 8 samples can be processed in parallel. Then the nuclei suspension is washed and filtered and optionally further cleaned by sorting on the max quant Tito cell sorter prior to single nucleus sequencing. Nuclei suspensions from different tissues will have different needs for cleanup. For example, the image on the left shows a nuclei suspension from mouse liver, and it is already mostly debris-free with just a filtration step. Alternatively, the image on the right shows a nuclei suspension from adult mouse brain, and it still has a considerable amount of debris even after filtering twice, first through a 100 micron strainer, then through a 30 micron strainer. The nuclei from the adult mouse brain would need further cleaning to remove debris before being used for single nucleus sequencing. To demonstrate the impact of cleaning nuclei suspensions, we took 2 frozen lung adenocarcinoma samples through our nuclei extraction workflow and then performed 10x single nuclei RNA-Seq on the nuclei suspensions both before and after sorting on the max quant Tito. We also used flow cytometry to quantify the level of debris both before and after sorting. We sorted on 7-AAD staining, which is a DNA binding dye, to separate the nuclei from debris. You can see here sample 1 was quite dirty. Only 9% of the events detected were nuclei before sorting. After sorting on the Tito, the percent nuclei increased to 40%, but more importantly, you can see we had a very considerable impact on library complexity. After sorting, the number of UMIs per cell nearly doubled, and the number of genes per cell increased by 50%. Sample 2 was much cleaner. It started largely debris-free, which is why the post-sorting improvement in library complexity is not as dramatic. This demonstrates that not all samples can be treated equally, especially when dealing with heterogeneous human tumor samples. Here are the resulting clusters from this experiment. We were able to identify cellular heterogeneity in the samples and were able to find the major cell types based on the expression of a number of established markers. One thing to consider when selecting a dissociation method for your single-cell sequencing experiment is that different methods will likely yield different proportions of cells. This is especially true when comparing cells to nuclei. A few papers have compared single cell to single nuclei sequencing of the same tissue and came to the same conclusions demonstrated here. While the same cell types were recovered with both methods, proportions were very different. Immune cells tend to be more prevalent in cell preps, whereas parenchymal cells tend to be more prevalent in nuclei preps. In this example, T cells, which are shown in black, made up around half of the cell prep and about 15% of the nuclei prep. Conversely, epithelial cells, which are shown in orange, made up about 15% of the cell prep and about half of the nuclei prep. The authors noted that while most protocols vary in cell recovery, it's not clear which, if any, provides the full ground truth of cellular composition. However, others have tried to answer this question. Let's take a moment to revisit this paper where they looked at every method. The authors analyzed cell type composition for single cell and single nucleus RNA-Seq as well as bulk RNA-Seq from their own experiment and another publication. For the bulk RNA-Seq, cell type composition was inferred based off the abundance of cell type specific transcripts. So the cell frequencies between methods were not drastically different. They didn't analyze immune cells in this graph. However, there is not a clear pattern pointing to the accuracy of one method over another. Let's look at neurons from adult mouse brain as one final example of the differences in cell proportions between cell and nuclei preps. In this experiment that we performed in-house, mouse brain was dissociated with the gentleMACS octodissociator with heaters and the adult brain dissociation kit. The cell suspension was then filtered with a 70 micron strainer, and red blood cells were lysed, and debris was removed with a debris removal solution. Finally, we used MAX technology and our adult neuron isolation kit to isolate the neurons. We then analyzed both the isolated neurons as well as the bulk unseparated cell suspension, both with flow cytometry and with 10x gene expression profiling. By both single-cell RNA-Seq and flow analysis, we found that only about 5% of the cells from adult mouse brain were neurons. In the single-cell RNA data, we were able to identify a number of diverse cell types, including microglia, astrocytes, oligodendrocytes, vascular cells, epidermal cells, and peripheral immune cells. We did find two small neuronal populations in the cluster graph. You can see them circled in red. And to further investigate the type of viable neurons that we were able to isolate with the adult brain dissociation kit, we then performed a second experiment where we first isolated the neurons. After isolation, neurons were enriched to over 90%. In the cluster graph again, all neuronal populations are circled in red. Because we were able to analyze significantly more neurons in this library, we were able to detect more neuronal heterogeneity. We identified 11 neuronal subpopulations, which consisted largely of immature neurons. As you can see, while we're able to isolate viable neurons from adult brain, we're not able to see the full neuronal diversity in adult brain because many neurons are too large and fragile to survive the dissociation process. This is why most researchers opt to isolate nuclei and perform single nucleus RNA-seq when investigating neurons. We also tested our nuclei isolation workflow by doing single nucleus RNA-seq on frozen adult mouse brain using the 10x gene expression profiling kit. Nuclei from adult brain tissue has a considerable amount of debris, so we also needed to use a max quant title microfluidic cell sorter to clear the nuclei from the debris. On the left, you see an image of the unsorted nuclei from adult mouse brain. We also analyzed these nuclei by flow cytometry and found that only 7% of the events detected were nuclei, meaning they were positive for 7-AAD. That means that the vast majority of the particles in that suspension were debris rather than nuclei. After sorting the nuclei in the max quant Taito, debris is no longer visible, and flow analysis determined that 90% of the events were in fact nuclei. When we analyzed the sorted nuclei suspensions from adult mouse brain, we saw considerable cellular diversity, detecting 34 clusters in all, as shown by the graph on the left. The two graphs on the right show the cells expressing a marker for either neurons or for a neural cell precursor. This demonstrates that nuclei preps are able to detect a significant amount of neuronal diversity, whereas neuronal diversity was limited when we used a cell prep. Okay, that completes all the methods that I have time to discuss today. But before we wrap up, let's summarize what we learned, and then I'll leave you with some practical advice. Every method has its own strengths and weaknesses, so hopefully I've gotten that point across today. There's never going to be one perfect method that does everything you want it to. So sometimes you have to make tough decisions. So let's say speed was really important to you. If that's the case, then extracting nuclei is a clear winner. Though if your nuclei suspension needs cleanup, this might change the situation. If you are working with very small amounts of tissue, then cell yield will be a big concern for you. In this case, you should try either nuclei isolation or warm enzymatic dissociation first. Let's say you're doing a large multi-center study, then protocol standardization may be important to you. In this case, your best bet would be to go with an automated kitting solution such as GentleMACS technology. So we currently have validated protocols for nuclei isolation and warm dissociation. So preservation of in vivo gene expression is important to everybody, I know, but it might be particularly important to you if you're investigating pathologies of cell activation. All four are good choices, but both by flash freezing intact tissue or putting intact tissue into fixative, you have the ability to lock in your cell state prior to dissociation. So those two would probably be the best options. You may need to enrich for or deplete a specific cell type from your sample prior to sequencing, and this is easily done with viable cells. Finally, the resulting cell type composition can vary between dissociation methods, especially between cells and nuclei. Single nucleus sequencing is better suited for very large or fragile cells. Whereas if you're really interested in cells with low RNA content, such as immune cells, they might get underrepresented by single nucleus RNA sequencing. So then you might want to go with cells. For fixed tissue, more study is needed, but early results show composition similar to warm cell dissociation. So I came up with some general rules, tips, practical advice for if you are just getting into single cell or single nucleus sequencing and are starting to develop your own sample prep methods. Preparation of target cells or nuclei is really the most critical step for a successful single cell sequencing experiment. Those experiments are expensive, so optimize your sample prep methods first, and that will save you time and money in sequencing in the end. And of course, if you can practice, definitely practice. I know people have issues with very precious tissue, but especially if you're working with mice, you have the tissue, practice before you start your experiment. Consider using flow cytometry as a QC method. It's fast and provides a wealth of information. I just always like putting this in here because I have a genomics background and I wasn't exposed to flow until I started at Miltenny, and it really is fast and there's a lot of information there. So if you are more of a molecular person, not used to the cell side, but now you have to start dissociating tissue and working with cells to do single cell sequencing, if you have access to flow cytometry, I definitely recommend it. So every dissociation method will have its own strengths and weaknesses. Try to be aware of them and then choose widely based on your tissue type, experimental needs, and cell types of interest. And then, of course, one dissociation method may not work well for every cell type in a tissue. Just be prepared that you might need different dissociation strategies if you want to analyze all cells in the same tissue. So I used a lot of literature references today. I just wanted to lay them out here for your convenience. And I also added a couple that were some good methods focused papers that has good information in it, but I did not have enough time to get to today. Thank you all for your attention today. I'll now be taking questions, and if you're watching this webinar later on demand, you could always email me a question. I've left my email up on the screen there, and if anyone would like a copy of the slides, feel free to email me as well. I'll send along a PDF copy. So thanks again, and I look forward to your questions.
Thank you, Carina, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many of your questions as we have time for. Okay, let's get started. Our first question is the gentleMACS compatible with cold dissociation?
Yes, so I actually showed an example of some researchers who did use the gentleMACS octo dissociator in a cold room for cold dissociation. So that seems to be the most popular way to do it. Just sticks the whole instrument in a cold room. I will mention that we don't have a validated dissociation kit or protocol for cold dissociation yet. It is something we're looking into, so it would be something that you'd have to figure out on your own. But the gentleMACS instruments can be operated in a cold room, and we will be releasing cooling units soon, so you don't need a cold room to do the cold dissociation if you wanted to figure out how to do that on your own. And of course, the nuclei extraction needs to be performed cold. So that's what the cooling units would be initially released for.
Okay, thank you. Question 2, how should I count nuclei?
Yeah, this seems like a really easy, obvious answer, but it's not. I think a lot of people struggle with counting nuclei because they use what they're used to, which is trypan blue on like maybe an automated cell counter. And nuclei with trypan blue on your typical automated cell counter just do not work really well. So obviously all the nuclei are going to stain blue, but all your debris is going to stain blue, and you tend to not get very accurate counts. So we really recommend using a fluorescent dye to count your nuclei, something that will bind the DNA and light up, so you get really good differentiation between what's nuclei and what's debris, and you'll get a much more accurate count that way. So fluorescent dyes can be like pretty much anything you want. 10x Genomics does recommend 7AAD if you're going to go do a downstream like a TAC-seq assay, since some of the dyes that bind to the DNA they found issues with. If you're just doing an RNA assay, then any DNA binding dye should be compatible, such as PI, DAPI, AIPO, whatever you have around.
Okay, thank you. Looks like we got a couple more questions here for you. Question 3, can I get a copy of the slides?
Yes, of course. So you can, to get a copy of the slides, I'll send everyone who wants one a PDF copy. You can just email me. So my email address is up on the screen right now, and I'll send those along.
Okay, thank you. Looks like we do have time for just one more question here, Carina. Question 4 will the Miltenyi dissociations kits work with transcriptional inhibitors?
So this is not something we have tested internally, but we've seen now several publications where people are adding transcriptional inhibitors to our enzyme digestion mix for the dissociation step, which is typically performed at 37 degrees, and have found that to help prevent dissociation-induced gene activation. So I can't say anything from Miltenyi because we haven't had a chance to validate it internally, but it does seem like some people are having success adding transcriptional inhibitors to the dissociation mixture, and it doesn't, from what I can gather, it doesn't seem to be inhibiting the efficiency of the dissociation. People are still getting the yield of cells that they're satisfied with. Yeah, I think it's really interesting, but I wish, we're working on it internally, so hopefully we'll have more to say later.
Okay, okay, great. Looks like we just got one late question here for you, and then we can wrap it up. Can we isolate nuclei from whole blood?
So from whole blood, I would recommend first removing the red blood cells. We do have a lot of products for working with whole blood. So the first thing you want to do is just lysol the red blood cells because that would obviously get in the way.
After you lyse the red blood cells and you're left with either your PBMCs or your buffy coat, yes, it's super easy to isolate nuclei from your white blood cells. So you don't need the gentleMACS for that. They are pretty easy to lyse. You can use our nuclei extraction buffer. Just sitting, incubating in the lysis buffer on ice is probably enough to lyse the PBMCs because they're fairly easy to lyse.
Okay. Well, thank you, Carina. Do you have any final comments for our audience before we go?
Yeah, so when I recorded this webinar, 10x had not yet released a protocol for FFPE on their fixed RNA profiling kit. However, they have recently released that protocol, and it does use the Miltenyi FFPE tissue dissociation kit upstream to dissociate the FFPE tissue. So this kind of just recently got released into the wild, so to say. So we're still waiting to hear back from how it's working in, the hands of everybody's researchers, but it's pretty exciting. So, keep following us, and hopefully we'll have more news for you about how that's working out. But it does seem to be a fairly simple workflow. Use our kit to dissociate the FFPE tissue. You do follow the 10x protocol. There is one key step difference. Luckily, that difference actually makes the protocol like about an hour shorter, so that's nice. And if anyone has any questions about that, you could, of course, contact Miltenyi or 10x.
Great. Thank you again, Carina, for your time today and your important research. We would also like to thank Labroots and our sponsor, Miltenyi Biotech, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. Labroots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.